Alright, so I've been talking about this a bit on our previous lectures, kind of leading up to it. I think chemistry and probably this membrane transport, these are two of the most important concepts or processes kind of in that physiological toolbox that you need to learn, because chemistry is going to be happening throughout the semester because all the cells work by chemistry, and this membrane transport will also be a thing that will be coming up throughout the semester, because all the cells are using chemistry and they're also going to be using membrane transport. So these concepts are important to get down so you don't have to keep relearning it every step of the way along the semester. Okay, so very important to get this knowledge down or these concepts down because it'll help your understanding and make your understanding easier in the future when we get to the organ systems or to the different organs and the different tissues. So if you remember, we've already talked about some of the fluids in the body, those solutions in the body. We have blood, we have this extracellular environment, we have interstitial fluid. Okay, the cells are bathed in a solution in this interstitial fluid or extracellular fluid. Okay, its extracellular environment includes everything outside the cell. You probably remember extracellular matrix. Okay, from many of your connective tissues has extensive extracellular matrix, but also there is usually a fluid component surrounding these cells, this interstitial fluid. Okay, and these cells, basically, if you remember the line of communication, stuff would come into the body, usually comes into the blood will pass to the interstitial fluid, and then from the interstitial fluid it can move into the cell, and then vice versa. We can go from the intracellular fluid inside the cell to the interstitial fluid to the blood, and then out the body. So we have to have this movement in and out of the cells. Okay, so you can kind of see that movement out, movement in, but we're going to be moving stuff in through the cell, and this is why we call it membrane transport, because our cell is surrounded by the plasma membrane. Okay, kind of like the boundary of the cell or the fence, keeping stuff in and out. Okay, so we get these movement across, and so that's where we have to come into play. Is we'll be spending most of the time talking about the transport across here between the interstitial fluid and the intracellular fluid. Here's just a visual representation. Okay, that's again showing the movement. We come from the outside, bring material in, bring it into the blood, bring it into the extracellular fluid and the interstitial fluid that surrounds the cell, and then into the cell. So just showing the movement through. What we're going to be focusing most on is this. Okay, the movement across here. This movement. Membrane transport across the membrane, across the cellular membrane. Right? And so this has to happen because, remember, this intercellular fluid's got to stay homeostatic. And to keep this homeostatic, we also got to keep these outside, the extracellular fluids, somewhat homeostatic, so we can get proper exchange between the cells and the extracellular fluid. Okay. All right, so since we're going to be talking about membrane transport, transporting stuff across the cell membrane, let's look what actually makes up the components of the cell membrane. Okay. We'll find cholesterol involved, but the main component is phospholipids, and I'll crudely draw one up here. If you remember, the phospholipid has a polar head and then tails that are the fatty acids, right? And in the cell membrane there is what we call lipid bilayer. There are two fatty acids wedged up, so it would just be a continuous strand of, here's one fatty acid, two fatty acids, three fatty acids, four fatty acids, but we would have this lipid bilayer Okay, continue all the way around and surrounding the whole cell. Phospholipids, the lipids portion. The phospho portion is polar, and this force is going to face out to the solution side, because this would be the extracellular fluid, and this would be, say, the inside of the cell, the intracellular fluid. Okay, this characteristic, these lipids here, give the characteristic of the membrane, the plasma membrane, as non-polar. Okay, non-polar characteristic. If you remember from our chemistry, remember polar things will dissolve in other polar things. Non-polar things will not dissolve in polar things, or won't dissolve very well. Okay, so keep that in mind. This plasma membrane that surrounds the cell, it has a non-polar characteristic. Okay, on these phospholipids, you could find cholesterol embedded in here. Might be between some of these. Also, you can see there might be carbohydrates that are tagged on the outside or inside. Okay, or also proteins could be tagged, and we're going to see for membrane transport proteins that actually span the whole thing. Okay, so the main component is phospholipids. You'll find cholesterol embedded in there. You also find carbohydrates attached to either inside or outside, and you also find proteins in there. Okay, but again, the main component is the phospholipids, this lipid bilayer. So here's a better rendition than what I drew on. Here is that polar head, all right, and the lipid tails. But you can see it's a bilayer all the way. This is the plasma membrane surrounding it. Okay, and you see there's some proteins embedded in. They don't have the cholesterol, but cholesterol can be in here, and also carbohydrates can be tagged out here. But remember, this membrane, the characteristic is, I know I've said it 100 times now already, but it is non-polar. Non-polar things like non-polar things. They play nice together. Polar doesn't like non-polar. Okay, again, the membrane is non-polar. So this makes the membrane selectively permeable. You can imagine polar things or charged molecules won't play nice with the membrane. They won't be able to intermingle and move their way through. Okay? Non-polar molecules, okay, can move their way through. So let's look at the permeability. Permeability, even though water is permeable, we will see size, besides polarity, size also makes a factor on if a molecule can move through the membrane or not. Okay, so we got polarity and size are going to be two factors that determine whether a molecule can move through the membrane by itself or not. Okay, water's kind of an exception. It is a smaller molecule. It is polar, but it can wiggle its way through the membrane. Okay, it's not the fastest, but it can wiggle its way through. Let's look at nonpolar molecules. Nonpolar molecules are non-charged. All right, and if they're small enough, okay, smaller is usually more likely to move through the membrane than very large molecules. And so we got a small nonpolar molecule, non-charged. All right, this can zip right through the membrane, okay, because it's nonpolar. All right, it plays nice with this nonpolar membrane, so it can find its way through the membrane directly. Okay, classic example of a nonpolar molecule that can zip through is cholesterol, and in turn, like progesterone and estrogen and testosterone, they are all made from cholesterol. Cholesterol is a precursor. Those molecules are small and nonpolar and can zip right through the membrane. What can't make it through? Large macromolecules, very big molecules aren't allowed in. Okay? Charged. What do we have for charged molecules? Remember our ions we talked about in chemistry. Okay? Charged molecules. Okay? Negative, that would be the chloride we've seen so far. Positive, that's sodium. They get shunted away. They can't make their way through. Okay? And then also polar molecules. Not shown here, but say glucose. Glucose is a polar molecule. It's not huge, but it's bigger than water, and it's polar. All right, it would also be shunted away. Okay, but we're going to see that we need to get these charged molecules. We have to move some in, and we have to move some out for our cells to operate. For glucose, you guys all know we need energy for our cells, and to get energy, we need some form of nutrient. Glucose is one of them, so we do need to get glucose in and out of the cell. Okay, so that's essentially what we're going to be talking about, how the cell is able to get these molecules in and out through this selective permeable membrane. So let's look at what kind of controls the movement across the plasma membrane. Okay, there are two requirements. We got physical and energy requirements that are going to control movement across the membrane. Physical requirements, how does it get across? How does it pass through the membrane? Okay, how we've seen with our nonpolar molecules, they can zip right through the phospholipid bilayer. Okay, so that's their requirement. They just have to go through the membrane. We will see for some of those other molecules that aren't allowed in directly through the phospholipid bilayer, we're going to see the use of membrane proteins or vesicles. Okay, depending on the molecule, it depends on how it can move across the membrane. Okay, the other component is energy requirements. Okay, sometimes the cell has to exert energy to move a molecule across the membrane. Sometimes it doesn't. Okay, so when we talk energy for our cell, we're going to use adenosine triphosphate, ATP. That is our fuel. That's our gasoline for our cells. Okay, we're going to spend a whole lecture talking about how this is made. But for now, if you see ATP, that means the cell is using it for energy. Okay, we're going to talk about active versus passive. Active would actually be using energy from the cell. Passive means the cell doesn't have to put energy in to make this movement, in this case, a movement of a molecule across the membrane. All right, so this kind of diagram here just walks us through pretty much what we're going to be talking about for most of this lecture. We saw there's two requirements involved in that membrane transport and the molecule passing through the membrane. We got our physical requirements, okay, whether it's going directly through the membrane here or if it's going to be mediated by a protein. Okay, we're going to have protein mediated transport. So those molecules that can't make it directly through the membrane, remember the membrane is non-polar, so polar molecules, larger polar molecules, have to be done by protein mediated. Okay, and then we'll look at even larger molecules. We'll look at membranes using vesicles. Okay, or sorry, movement using vesicles. The other, we saw energy requirements. Some movements don't require energy, others are requiring energy. Okay, and so the first one we're really going to talk about, diffusion. Okay, it can be simple or facilitated. Okay, and then we are going to look at active movement. Okay, and we're going to see primary, secondary across the membrane, and then the larger stuff we'll see the endocytosis, exocytosis, and phagocytosis. Okay, so first we're going to look at basically passive movement. We're going to look at diffusion. Okay, how do molecules diffuse? And so this being passive, it does not require energy from the cell. Okay, it does not require that ATP, that gasoline, to move the molecules through the membrane. So since we're going to be talking about basically we're looking at movement in or out of the cell through the plasma membrane, you got to remember that the cell is bathed by a solution and the cell itself has a solution in it. So it has a solution. It has a solvent with molecules or solutes dissolved in it. And one of the characteristics we got to look at is concentration of a solution. And that's basically measured in what we call molarity or molality. Okay, this tells you the concentration. And I'm not going to get big onto the mathematics of it, but what I want you to realize is molarity and molality are measures of concentrations. Okay, this is telling you how much solute is dissolved in the solution. Okay, and again, we have a different solution outside than we do inside because it's separated by that plasma membrane. Okay, but one of the driving things for what we're talking about diffusion is we have to take into effect concentration. All right, molarity, I can go through, show you how it's taken into account, but what I want you to realize is 1 molar solution of, say, glucose is half as much as a 2 molar solution. All right, so there are twice as many glucose molecules dissolved in this 2 molar solution than there is in this 1 molar solution. Okay, and typically what you do is you look at a mole dissolved in a liter of water. So this essentially would have 2 moles dissolved in a liter of water, where this would only have 1 mole. Okay, so this has twice as many molecules of glucose in the same amount of fluid. It's 1 liter of water. In our case, we're using water as a solvent. Okay, so you can see that's what molarity is telling you. It's telling you how concentrated it is. This one is twice as concentrated as this. There are twice as many glucose molecules in the same amount of water, same amount of solution as there is in a 1 molar solution. Okay, because what we're going to be doing is comparing inside to outside, and that's going to allow us to figure out where a molecule will move. And this is just kind of give you a visual representation. Molarity and molality, because we're using the body, there's not a huge difference between these, so I'm not going to get into that. Typically, you're going to see molarity used, but you might also see molality. But in the solutions that we're using in the body, these are very closely almost the same. Okay, there's not a huge difference between a molarity and a molality solution. But what I want you to look at is here. Okay, here's 1 liter of water. We're putting in 1 mole of glucose. That's 180 grams of glucose in and dissolving it. For a 2 molar solution, we would pick 2 moles. We would put 360 grams into the same amount of water, this 1 liter solution. So that's just telling you that there is twice as much solutes, twice as much glucose molecules in that 2 molar compared to that 1 molar. Okay, and that's essentially what molarity is telling you. So the reason we care about this molarity, this concentration, what is the concentration of a solution, is because our first process, this diffusion, okay, this is a passive process in which the cell does not have to put energy to move a molecule either out of the cell or into the cell through the plasma membrane. Okay, and the key with diffusion is
You got to remember these solutions are solutes, are dissolved in here. These solutes are these little particles. Okay, and they're just not sitting there in the solution, sitting stagnant. They have what's called kinetic energy. They're moving around, bouncing around. Okay, and these molecules with this kinetic energy, they don't want to stay concentrated next to each other. They want to have distance, as much distance as possible. So if looking here, the simple part of diffusion, if we put a cube of dye into water, what happens with the dye? It doesn't sit concentrated in one inner area. It wants these dye molecules, these dye solutes, won't stay here. The kinetic energy moves them from an area of high concentration to an area of lower concentration. There is no dye up here, so the dye starts moving out into these areas where there is no dye of low concentration, okay, until we reach what we call equilibrium. Okay, so the main thrust of diffusion is that a molecule, a solute, will move from an area of high concentration to an area of low concentration of the same solute. Okay, and this is called movement down the concentration gradient. Moving from high to low is down. We will see another instance where you can move from low to high, and that's moving up the concentration gradient. Okay, so some of the main features. Diffusion, we're going from high to low. The energy is coming from the molecule itself. It's in its kinetic energy, that bouncing movement of the molecule, inherent in the molecule. Okay, and it also will move, if it's left to go, it will move, the molecules will move until they reach equilibrium. So this is just kind of a getting a written definition of what I just talked about. The kinetic energy is the movement, is the drawing force, is where the energy comes from. It's kinetic energy in the molecule, diffused from high to low concentration, and diffusion will occur until it reaches equilibrium. Some factors that might affect diffusion are diffusion is faster along higher concentration gradients. So if there's a lot more, say there's 3 molar solution versus 0 molar, it's going to move, the molecules are going to move faster. But what if there's a solution with 2 and you put it next to a solution with 1 and then add those together? The 2, it will move, because there's only a difference from 2 to 1, the concentration difference is smaller, and therefore the molecules won't move as fast. Diffusion also occurs faster over shorter distances. Higher temperatures increase diffusion. Remember, what's the driving force for diffusion? It's kinetic energy. And at higher temperatures, those molecules will be bouncing faster, much quicker, and therefore diffusion is much faster. Okay, and then smaller molecules move faster than these big clunky molecules. Therefore, smaller molecules move faster. The diffusion of smaller molecules will also move faster. You kind of saw that in your lab. You saw the methylene blue, which much heavier, much bigger than the potassium permanganate. Potassium permanganate diffused much faster because it is a smaller molecule. Right, and this diffusion can take place in an open system, or what we're going to be mainly focused on is across a partition that separates the two systems. In this case, the two systems is the interstitial fluid on the outside of the cell, and the partition would be the plasma membrane, and the other solution would be that solution that is inside the cell, the intracellular fluid. So on our map of membrane transport, where we're going to head to first is simple diffusion. Okay. All the properties of diffusion apply. We're moving across. We're moving. So it's diffusion. So the molecule is gonna move from high to low. It's not gonna move from low to high. Okay, that's the driving force. Is the kinetic energy. All right, and now we're going through a membrane. In this case, simple diffusion means the molecule is passing directly through the plasma membrane. Okay, it's going through the lipid bilayer. So what kind of molecules do you think this is? We kind of saw that earlier map. We saw small molecules move through the plasma membrane easier, but also what other characteristic is important? Nonpolar is more likely to move through the plasma membrane than polar molecules. Although we saw the small water molecule is able to make it through the plasma membrane, but small polar molecules can go directly through. But again, there has to be a concentration differences between the outside and the inside of the cell, because we must have a concentration difference for diffusion to occur. And again, the molecules are going to move from area of high concentration to an area of low concentration. All right, so here we look at simple diffusion. Simple diffusion is taking place directly through the plasma membrane. And again, it's small molecules are more likely to move through, and also molecules that are lipid soluble or that are nonpolar are more likely to move through the plasma membrane, where polar molecules will get rejected away. Okay, but again, simple diffusion means it's moving directly through the plasma membrane. But again, if the molecule is moving from here to here, what side must we have a higher concentration? Yeah, we have to have the higher concentration would have to be on this side over here, because the molecule's got to be moving, because of diffusion, high to low. So there's a lower concentration of this molecule on this side and a higher concentration on that side. So we've already seen properties that affect general properties of fusion, but here are some that are going to affect the rate of diffusion across a membrane. Okay, membrane surface area is larger. So the larger surface area, the more diffusion that takes place. You remember in anatomy, we saw in digestive tract. A digestive tract, when we absorb, we're diffusing nutrients and so forth in from the digestive tract into the body. And we saw all these folds in the lining of the digestive tract because that gave that area a larger surface area. So surface area, if it's larger, we can move stuff in more rapidly. Besides being larger, if we have a large surface area and a thin surface, we're also able to move stuff in faster. And then if the concentration gradient is larger, again, if you have tons of stuff, so you will see our cells, we have lots of sodium outside the cell and very little sodium inside the cell. And so the diffusion across there will take place much, much faster. And the membrane is more permeable to the molecule. And what makes the membrane more permeable is molecules' lipid solubility. In case of the plasma membrane, the more nonpolar the molecule is, the more likely it's going to move through. But also size has a factor, and also the composition of the membrane. We won't look so much at this, but we'll see these two factors up here come into play when we're talking about molecules moving across the membrane. All right, so we've seen simple diffusion. Simple diffusion essentially is a solute moving from across the membrane from an area of high concentration to an area of low concentration. So simple diffusion, typically the molecule is a smaller nonpolar molecule, but we did see water can move across the membrane, and there is a term for this. Water moving from an area of high concentration to low is osmosis. Water movement is osmosis. Okay? And water will move from an area of higher concentration of water to an area of lower concentration of water. Okay? And there are two requirements for osmosis. There's got to be a difference in concentration of solute or solutes, combination of solutes, on the two sides of the membrane. And the membrane must be impermeable to some solute or all the solutes, but there must be impermeable, or what we call non-penetrating solutes. Okay? If they are non-permeable or non-penetrating, that means the solutes cannot move across the membrane. In this case, it's the purple solutes, and the water are the little blue. Okay? These solutes are termed, the ones that are non-penetrating or non-permeable are osmotically active solutes, because they are going to determine which way water goes, where the movement's going to take place. And so I did say osmosis is, in a sense, a movement, a diffusion. You got to think that water, if solutes are highly concentrated in a solution, there's less room for water. There's lots of solutes in there. There's less room for water. So that a high solute solution would be a low concentration in water, where a low solute concentration solution would have a higher concentration of water. So you got to think of it almost in reverse. More solutes means less water. Okay, so in that case, solutes will move from high to low, but in this case, water is actually going to move from the low solute concentration to the area of higher solute concentration. But the importance is also you have to take into account the solutes. The solutes must be impermeable. So water is going to move from an area of water is going to move from an area of low non-permeable solutes to an area of high non-permeable solutes or non-penetrating solutes. Okay, so I know that this is the concept that usually gets kind of mixed up. I'm going to try to find you guys some resources too so you can watch outside. But if you're also having trouble, come see me in office hours, because this concept kind of gets mixed up. But remember, osmosis is diffusion, so water's moving from high water to low water. And it's kind of vice versa of what the actual concentration of the solutes is. So we've looked at molarity. You've seen molarity had to do with what solutes were dissolved. So if I put one glucose molecule, 1 mole of glucose in 1 liter, that gave me a 1 molar solution of glucose. If I took 2 moles of glucose and put it into 1 liter of water, that gave me a 2 molar solution. Okay, for osmolarity, for osmosis, we have to use what's called osmolarity. We have to add a little bit to this molarity portion. Okay, because we have to, osmolarity cares about, or osmosis cares about, the osmotically active particles per liter of solution. Okay, and this changes between, say, a 1 molar solution of NaCl versus a 1 molar solution of glucose. Okay? Because what we're talking about is not molecules, we're talking about particles. And remember what happens, do you guys remember what happens in chemistry when you put NaCl in a solution in water? water dissociates. So you actually end up with, when you put NaCl in water, you end up with a sodium ion and a chloride ion. That is 2 particles. Okay, so for ionic molecules, ones that will dissociate in water, you have to multiply by the number of particles that is made. In this case, we got 2. So a 1 molar solution of NaCl is actually 2 osmoles, because it's the number of particles for osmolarity. Where glucose is a covalently bonded molecule. Glucose doesn't break up. It doesn't get dissociated in water. So the glucose molecule still stays intact as 1 glucose molecule, so it stays as 1 particle. And therefore 1 molar glucose actually equals 1 osmoles. So osmolarity-wise, even though molarity-wise NaCl and glucose are the same, osmolarity-wise they are not. Okay, this is a big difference when we're talking about movement of water, because movement of water is going to be determined somewhat on osmolarity. Okay, we will look in our cells. Okay, because we're going to be looking at movement across the plasma membrane. So we care about what the osmolarity, because osmolarity is going to dictate water movement. So we care about the difference in osmolarity between the inside of the cell and the outside of the cell. For human cells, it's roughly actually 297, but we can use 300 milliosmoles, again, of non-penetrating solutes. Okay, I'll make the distinction again that it is the non-penetrating solutes that cause the movement of water. Okay, they are the osmotically active solutes. So there's conversion. We got a number of particles formed in solution. Again, ionic compounds dissociate, so they form more than 1 particle, but covalently bonded molecules only form 1. Okay, so we can take comparisons between solutions. We're going to be comparing because we're going to look at osmolarity, where water is going to move, if water is going to move inside the cell or move outside the cell. Okay, and that's going to be determined by the osmolarity of the solutions inside or out of the cells. So we got to kind of compare solutions. And we can compare solutions, and we have some terminology when we do this. Okay, solution A is 1 osmole of glucose. Solution B is 2 osmoles of glucose, and solution C is 1 osmole. Okay, this has already been converted to osmoles. It's not molarity, so this is 1 osmole of NaCl. It is not 2 because we don't have 1 molar NaCl. This would actually be 0.5 molar solution of NaCl, because we're going to times 2 times 0.5 equals our 1 osmoles. Okay, but let's just compare these different solutions. If we compared solution A to solution B, which one is lower? 1 osmole is lower than 2 osmoles. So solution A is considered what we call hypoosmotic, 2B. A and C, 1 osmole and 1 osmole, it is isoosmotic. Iso meaning same or similar. Okay. So if it's below, it's lower in osmolarity, it's hypo. If it's same, it's iso. And let's look at solution B compared to A. 2 compared to 1. 2 is higher than 1, so B is considered what we call hyperosmotic, above osmolarity, above the osmolarity of solution A. And B is also hyperosmotic to C because C is 1 osmol. Okay, and then the vice versa, C versus A, 1 and 1, they're iso, and C is lower than B, so it is hypoosmotic. You're comparing solutions. Okay, it depends on how you're comparing one to the other. Okay. So we're going to take this terminology and then we're going to actually add to it too when we start talking osmolarity and tonicity. We're going to add another term to this called tonicity. So let's look at osmosis and kind of the process involved. We have sucrose. Sucrose is here, dissolved in water. I have 180 grams of sucrose per liter in the outside. The inside, the dialysis bag here, it's 360 grams per liter of sucrose. Okay. The key is this semi-permeable membrane sac, the sac here, is not permeable to sucrose, but it is permeable to water. So water can move in and out, but sucrose cannot. This is important because remember the solutes. We care the osmotically active solutes, the one that are going to drive water one way or the other, are the ones that are the non-permeable solutes. In this case, sucrose cannot penetrate or is non-permeable to the cell, so it's not moving in and out. Okay, so it is one of those osmotically active solutes. There's twice as much sucrose as there is sucrose in this solution outside the sac. So which way is water going to move? Water is going to move to the area of, there's higher concentration of water here and lower concentration of water there. So water is going to move into the sac. Okay? There is higher osmotic pressure in this solution. Higher osmotic pressure is going to drive the water in to the solution. Okay? Again, water is going to move to an area of higher non-permeable solute concentration solution compared to the lower concentration. Lower here, higher in here, so water moves in. So we can look at a quantitative measure of osmosis, and that's what we call the osmotic pressure. So a solution, if it draws water in, it has a higher osmotic pressure. Okay, and there's a pressure that must be opposed to prevent the movement of water. So if we had a solution here, 2 compartments, selectively permeable, this right here is permeable to water, but not to the purple solutes. And as you can see, there is higher concentration of solutes, non-permeable solutes, non-penetrating solutes on this side compared to this side. Okay, again, remember it's the non-penetrating solutes that are gonna draw the water. And water is going to be drawn into a higher concentration solute, higher osmolarity. And so water is going to be drawn in this. So if we can measure osmotic pressure is basically how much pressure has to be pushed here to keep the water columns equal, because water is drawn into this higher osmotic pressure. So solution. Okay? So what's the pressure we got to put on? That's how you calculate the osmotic pressure. Why do we care about this osmolarity? We care because there's another terminology called tonicity, and this affects our cells. Okay, because remember, we're looking at water moving. What we're caring about is water moving in or out of the cells. Okay, so if we move water out of the cells, it's going to affect our cell volume. Or if we move water into the cell, it's also going to affect our cell volume. Our cells like to stay nice and same volume. Okay, typically we use red blood cells in these experiments where we're looking at the effect of tonicity on a cell. Okay, but what does osmolarity have to do with this? Well, osmolarity is somewhat the driving factor in driving water in or out of the cell. Okay, those osmotically active solutes, if there's more in the cell than outside, then water will be pushed into the cell. If there's more osmotically active solutes outside the cell than inside, the water is going to be drawn out of the cell. Okay, how a solution affects the structure or the tone of the cell is what we term tonicity. Okay, tonicity doesn't have any units. We're not comparing, we're just looking at what a solution will do to the tone of the cell. Okay, and what I want you guys to remember is there's 300 milliosmoles of non-penetrating solutes inside the cell. Okay, so let's look at first an isotonic solution, same tone. Right? If I have a solution that has 300 milli, the solution here has 300 milliosmoles of non-permeable solutes out here. Okay, and there's 300 non-permeable solutes inside the cell. The amount of non-permeable solutes outside and inside are the same, so water will not be drawn out and won't be drawn in. There won't be a net flux of water in or out of the cell, and therefore the cell's tone stays the same. Little biconcave disc. That is an isotonic solution. So let's say our solution out here had 400 milliosmoles. And what did our cells have? Our cells had 300 milliosmoles. In that case, there is more solutes outside than inside. And where's water going to be drawn? It's going to be drawn to the higher non-permeable osmolarity solution outside. And when a solution shrivels the cell, or what we call creates it, okay, it is a hypertonic solution. So hypertonic solutions shrivel the cells. On the opposite end, what if I had 100 milliosmol solution that this cell is bathed in? Now, where is my higher ion concentration? It's inside the cell, right? So now water is going to be drawn to inside the cell. And this is an example of a hypotonic solution. A hypotonic solution is going to drive the water into the cell and swell it. Eventually, if there's enough, it's going to lyse the cell. Okay, so that is tonicity. Tonicity just has to do with what's the effect on the cell. If it swells it, it's hypo. If it shrivels it, it's hypertonic. And if the cells stay nice and happy and the same, it is an isotonic solution. But it is drawn by the concentration of ions, specifically the non-penetrating or non-permeable solutes. The ones that won't move through the membranes are what driving whether water will move in or out of the solution, or sorry, in and out of the cell in this case. So tonicity and osmolarity are related, but they're not always the same. Osmolarity, basically we're comparing osmolarity between two solutions. When we compare the solution compared to the osmolarity in the cell, we're comparing the 300 milliosmoles in the cell compared to what the osmolarity is outside. So osmolarity has units. When we talk tonicity, okay, tells us what's happening to the cell. Tonicity compares the solution to what is happening to the cell. Is the cell shriveling? Is the cell staying the same? Or is the cell swelling? Okay, so there's no units there, but it's telling us what's happening to the cell. So that's what tonicity is. Okay. And tonicity is related to osmolarity because osmolarity is going to be a drawing factor for water, but also osmolarity has to do with the total amount of solutes in a solution. It doesn't tell you whether solutes are non-penetrating or penetrating. Okay, remember we only care about the non-penetrating solutes and for osmolarity or for osmosis. Okay, so for tonicity's sake, all we really need to compare is the non-penetrating solutes inside or outside the cell. That is going to give us what effect is going to happen in the cell. Okay, this is so slow. Penetrating. Okay, non-penetrating solute. I'm going to actually write it here because this is the key characteristic, non-penetrating. How many non-penetrating solutes are inside compared to outside the cell? And that's going to tell you where the water is going to move. The water is going to move to a solution that has a higher amount of non-penetrating solutes. Okay, and that's going to tell you the tonicity, because wherever water moves tells you if water's moving in, it's a hypotonic solution. If water's moving out of the cell, it's a hypertonic. Okay, and if water's not moving in or out, no net flux of water, then it is an isotonic solution. Okay, and why do we care about this? In health field, you're going to be using IVs. IVs contain solutions that you are going to be putting into a body, into a person's blood. Okay, and this, in effect, we're putting in osmolarity. We're putting in what is the osmolarity of that IV is going to be important to the patient. So let's look at the factors that affect tonicity of the solution. All right, we've already said osmolarity is one of those factors. Okay, but osmolarity only tells you the number of solutes or solute particles that are in the solution. It doesn't tell you the nature of the solutes. And remember, said it many times now, but it is the non-penetrating solutes that we care about because these are the ones that are going to determine where water moves. Okay, so non-penetrating solutes, 300 milliosmoles in the human cell, roughly around there. That's what the human cells keep in there, is 300 milliosmoles. Okay, so essentially to determine tonicity, or to determine where water is going to move, all we have to do is compare non-penetrating solutes, the osmolarity of non-penetrating solutes outside or in the solution compared to the osmolarity of non-penetrating solutes in the cell. Okay, so let's look here. When the concentration of non-penetrating solutes in the cell is equal to the solution, so the solution has 300 milliosmoles, again, of non-penetrating solutes. The cell and the solution are equal, and therefore water will not move, and therefore it is isotonic. Now, what if the cell has a greater amount of non-penetrating solutes than the solution? So the solution is lower in non-penetrating solutes. So let's use a lower case. Lower, so in this case up here, the solution is 200 milliosmoles. Okay, where is the water going to move? It's, in this case, the water is going to move to the area of higher non-penetrating solutes, and that would be in the cell in this case, and that would increase the cell volume. And remember, if you increase the cell volume, the tonicity is hypotonic. And then finally, what if the cell has a lower amount of non-penetrating solutes compared to the solution? In this case, so the solution has more. So the solution would, say, have 400 milliosmoles. Okay. So where would the water move in this solution? The water would move from area of lower non-penetrating solutes to an area of higher non-penetrating solutes. In this case, it's the solution. So water moves out of the cell, and if water moves out of the cell, it's considered a hypertonic solution. So again, the take-home message here is just compare non-penetrating solutes inside the cell compared to non-penetrating solutes outside the cell, and that is going to tell you where water is moving, and in turn can give you what the tonicity is. Okay, so we're just kind of repeating what I'm saying, but I'm going to still hit home. It is the non-penetrating solutes. And what we're going to find in physiology, the most important non-penetrating solute out there, okay, is sodium chloride, specifically sodium. Sodium, you can think of, is where sodium goes, water is also going to go, because sodium is a non-penetrating solute. It's the most important one in physiology when we find solutions. Sodium chloride, okay, the salt, remember these are ions, so they are charged, so they are not going to move through that non-polar membrane. Okay, we will see there's cases where they can, but they cannot move directly through the membrane. Okay, so they are considered non-penetrating solute, and so it is the most important. And so why do we care about this tonicity? You saw tonicity affects the cell's shape. Would you really want your cells shriveling up, or would you want your cells swelling to a point where they could almost burst and break open? No. So we want to keep our cells nice and happy in health. We're going to be using IVs. We're putting IVs. And it's important that the osmolarity of those IVs match so we can keep our cells nice and happy. Because if we add too much salt or too little salt, too much non-penetrating solutes, then we can affect the tonicity of the cell. We can expand the cell or shrivel it, and that's going to affect the activity or the functioning of the cell. So it's important that the tonicity of those solutions that we are putting in are matching what we need to do in the body. This is a good rule of thumb, but mainly the main thing that's taking place here is we have to determine non-penetrating solutes. Okay, you're going to get some examples of questions. You're going to be given osmolarity solutions, and then you're going to see you can have a hyperosmotic solution. All right, you can put your cell in a solution that has 500 milliosmoles, right? And that solution can stay perfectly fine. Okay, let's see why that is the case. So looking here, we can look at osmolarity. Okay, and we're looking at tonicity. What's the effect on the cell? Remember the magic number for the cell is, magic number for the cell is 300 milliosmoles. Okay, 300. So let's look up here at always hypoosmotic cells, cells that are lower than 300, 200, 100 milliosmoles. They will always be hypotonic. They will always, in hypotonic, they will increase water into the cells. They will swell the cells. But let's look at isoosmotic. You can have an isoosmotic solution, and everybody thinks, oh, it should be isotonic, but it can also be hypotonic. Okay? Isoosmotic just means the same osmolarity. In this case, what would isoosmotic mean? The cell is 300 milliosmoles. So what if this solution was 300 milliosmoles? Okay, this solution's 300. But what is that osmolarity telling you? It's just the number of particles. It's not telling you the nature of the solution or the solutes. Okay? What do we care about when we talk about tonicity? What is going to move the water? What kind of solute? Again, it's the non-penetrating solutes. So I can have a solution that's 300 milliosmoles, but if 100 of those milliosmoles is devoted to a penetrating solute, one that can move through the membrane, then only 200 milliosmoles is non-penetrating. Again, what do you compare? You compare non-penetrating in this solution and non-penetrating in the cell, and where's the water going to move? It's going to move into the cell. Okay, again, it's just stressing that it is the non-penetrating solutes that result in water movement and that are involved in tonicity. Let's go here. We got hyperosmotic. Of course, if we have a 400 milliosmol solution and all of those are non-penetrating, then the water is going to move from the cell into the solution. Okay, but we could have 400 milliosmoles, but 100 of those milliosmoles can be penetrating, so that would leave us with 300 non-penetrating, and therefore then we would be at isotonic. Again, if we have a 400 milliosmol solution, but 200 of the 400 milliosmoles are devoted to penetrating solutes, that leaves me only with 200 non-penetrating solutes. If we compare 200 to 300, then water's going to move in there, and that makes the solution hypotonic. Okay, so again, this graph is just basically stressing the non-penetrating solutes are the ones that are the most important, or that are going to be the only ones causing the water movement.
So from a medical standpoint, tonicity is a very important concept because many times in the hospital or even outpatient, IVs are put into patients. IVs are putting fluids directly into the blood, directly into the extracellular fluid.
Okay, so the osmolarity and tonicity, or really the tonicity of this solution, is important because it's going to affect the tone of the cells. And we can do several things. We can have, if we have dehydrated cells, we can use a hypotonic solution, because remember, hypotonic solutions are going to increase water into the cell. Or in cases of like blood loss here, we can use isotonic. Isotonic because we don't want to change the cell. We've lost blood. We just need to bring in fluid. We want an isotonic solution. So looking at some of the common solutions, IV solutions, we can look at one known as normal saline. Saline, remember, NaCl. Saline is a solution of water and salt, NaCl. Okay. That is what saline stands for. Remember, NaCl is the most prevalent or most important non-penetrating solute. Again, non-penetrating solute. So a 0.9% solution of saline is called normal saline. And this solution is isoosmotic. Isoosmotic, remember the cell is what? 300 milliosmoles. 300 milliosmoles of non-penetrating solutes in the cell. So what do you think? If this solution is isoosmotic to the cell, what should its osmolarity be? Yeah, it's got to be 300. And if you look at this, saline, remember saline, NaCl, is a non-penetrating solute. So if we go to compare non-penetrating solutes in the solution and non-penetrating solutes in the cell, it's 300 to 300. So is water going to move? No, it makes this solution isotonic. Let's look at further down. 5% dextrose in 9.9% saline, normal saline. I will let you know that dextrose, 5% dextrose, if you calculated it, if you did the osmolarity calculation, it would be 300 milliosmoles. Okay? But the dextrose is in with normal saline, which also adds 300 milliosmoles to the solution. So in regards, this solution here has a total of 600 milliosmoles. So if you compare osmolarity of this solution to the cell, 600 compared to 300, it makes the solution hyperosmotic. But I will let you know you can consider dextrose as non-penetrating. Dextrose is non-penetrating. Again, when we talk tonicity, what do we care about? When we talk tonicity, all we care about is the non-penetrating solutes. The non-penetrating solutes of a solution is going to tell you what's going to happen with water. And in this solution, 5% dextrose is penetrating. So we can just disregard this 300. When we're talking tonicity, we don't have to worry about penetrating solutes. What we do need to worry about are the non-penetrating solutes. In this solution, we have 300. We compare 300 to 300 in the cell, and that ends us up with an isotonic solution. Okay. Let's go down. I'll go down to, well, we'll do 5% dextrose in water. Okay, I have already told you that the dextrose is 300 milliosmoles if you calculate it. If you put a cell, so let's 300 dextrose, 300 milliosmoles, that makes the solution isoosmotic. Okay, but tonicity wise, Remember, dextrose is penetrating. So we have dextrose in water. How many non-penetrating solutes do we have in this solution? Do we have any saline? We don't have any saline. So this actually has 0 non-penetrating solutes. And what do we care about for tonicity? We care about the non-penetrating solutes. So if we compare 0 to 300, the water is going to move towards into the cell. Okay, and that makes the solution then hypotonic. A hypotonic solution, water moves into the cell. Okay, so this goes towards the tonicity. You don't want to put a gob amount of, a hypotonic solution. If a cell is, if you've got a dehydrated person, you might want to put some hypotonic solution in to puff up the cells to bring them back, bring the hydration back. Okay, but you can kind of work through these looking at, but again, the point is it's the non-penetrating solutes that are the ones that we are concerned with. Okay, back at the other side, I talked about look at the solution before you don't think about the water movement, just what is the osmolarity, compare the osmolarities, and then you can figure out which way the water moves, and you can figure out the tonicity of the solution. Okay. So we've finished up with movement through the membrane. Water can move through the membrane, the plasma membrane. Those small nonpolar molecules can move through the membrane. So we finished up with our simple diffusion, things that can go directly through the membrane. Now let's talk about stuff like glucose, okay, or sodium ions. Right. We're also going to talk a lot about, we haven't talked potassium, but potassium's the big K ion. These molecules, they're charged, even though sodium and potassium are small, they are charged, or glucose, which is larger and polar, they can't make their way through the membrane. They're not allowed to go through. Okay, so how do we solve this problem? This way that problem is solved is we use proteins. There are proteins that span the membrane. Okay, they span the whole membrane, so the outer part of the protein can face the extracellular fluid, and the inner part of the protein can face into the intracellular fluid inside the cell. These proteins span, and they act as transporters for these charged molecules, larger nonpolar molecules. So this is considered protein-mediated transport. Okay, we can do a form of diffusion, facilitated diffusion, and we can also use energy to move molecules through. And we're going to look at the difference between these, but first we'll take a look at what's called facilitated diffusion. And just look at the word first is we got diffusion. Diffusion, when you ever see diffusion, it means a molecule, a solute, is moving from an area of high concentration to an area of low concentration. When you see diffusion, you need to think high to low. And in that case, where is the energy coming from? It's coming from the kinetic energy of the molecule. Okay, those molecules are bouncing around. Okay, but the kinetic energy will never move the molecule from an area of low concentration to an area of high concentration. Kinetic energy will move the molecule from high concentration to low concentration, causing the diffusion. Okay, and in that case, the cell does not have to use energy. So just looking closer, again, we're going to touch on facilitated first, but let's look at the difference between facilitated diffusion and these other two, these active transport. Okay, and what does passive and active? Facilitated diffusion is passive. We already said the molecule moves down the concentration gradient, moves from high concentration to area of low concentration. So the cell does not have to push energy or use energy to make it work. The energy comes from the kinetic energy in the molecule. Where's active transport? This is where we differ. It requires energy from the cell. A cell must use ATP or another source of energy to make the molecule move through the membrane. And the reason for this is because in active transport, the molecules are not, or the solutes are not moving down the concentration gradient. They are not moving from high concentration to low concentration. In active transport, the solute is moving against, saying it's moving from an area of low concentration to an area of high concentration. So kinetic energy of the molecule cannot be used, so the cell has to actively transport the molecule to go from low concentration to high concentration. Okay, so whenever you see active transport, the molecule is going against the concentration gradient, going from low to high. Whenever you see the word diffusion, all right, it means the solute is going from an area of high concentration to an area of low concentration. All right, so before we actually get looking at the movement, the facilitated diffusion, and the active transport, let's look at the membranes that are going to be used to make these types of transports happen. We have two types. We got carrier proteins, all right, and you can see they can change conformation, and we'll talk about this in a bit. And then we have channel proteins that also will allow molecules to move through the plasma membrane. Again, we're talking about larger polar or charged molecules that are being used to move through the membrane, because those small nonpolar can move directly through. Okay, so we're going to use these membrane transporter. These are proteins embedded in the plasma membrane. So we'll go through these, look at their characteristics, and then we'll move into the actual movement of the solutes through these proteins. So here we look at the two players in membrane transport. Okay, we have channels and we have carriers. Okay, if you're looking at the channels down here, this is a channel. It basically, you can see both of these actually span this membrane. They got to have connections between the outside, the ECF, and the inside, the ICF fluids. Okay, but the channels are more like they're a water-filled passage, but they only allow certain molecules. They're usually specific for a certain molecule or a certain type of molecule. Not everything can just flow through here. There are characteristics that make it so on some only a sodium ion can move through. Okay, so it's not just everything can come through. It just depends on the channel and what would be allowed to come through here. Okay, but the ICF and the ECF, okay, the inside and outside, basically it's like more like opening a door. They have access to each other. Where carrier will change conformation. Again, these are proteins, so they can change their shape. Here, you can see the carrier is open to the ICF, but then when it changes shapes, it's open to the ECF, but not the ICF. Okay, so it's more like a revolving door. It's not like there is direct access between the extracellular fluid and the intracellular fluid. Okay. This gives some characteristics. This also involves that the molecule or the solute has to bind. Usually it's the binding that's going to change the conformational shape, or ATP can change the conformational shape, but the molecule in the carrier has to bind. Whereas here, the molecules can just flow through. So this is much quicker than, say, carrier movement. We'll see that later on when we look at the characteristics of these. So looking at our channels, okay, you see the membrane channels here. They span the membrane, and molecules can move just through basically this tunnel, this pore, through the ECF to the ICF. Okay, what's channels? Channels usually only handle small molecules. Okay, bigger stuff like glucose and so forth, we're going to have to use those carrier proteins to move molecules through. But again, channels, small molecules, so we're talking ions. You will hear them as ion channels, so small ions. There are also channels for our small molecule water. We already saw that water can move through the membrane, but water has to kind of wiggle through, and it's slow. If we needed to move water very quickly in and out of the cell, we can have a channel called an aquaporin that would allow water to rush through. Okay. The other thing about channels is there's open channels. So there are channels that are open all the time. Okay, they are never closed, so molecules are always moving in and out. But there are also gated channels. Okay, gated channels can be closed or open. Okay, and usually it is a queue. There is a queue that opens up these channels. Okay, it can be chemically, voltage, or mechanically gated. The other thing I want you to realize on channels is there is no active transport taking place through channels. Channel proteins utilize passive transport. So passive, when we hear the word passive transport, what are we talking about? We're talking about diffusion, right? So only diffusion can take place through a channel. Okay, we will see later carrier proteins, they can do diffusion, but they can also cause active transport. Okay, but for channel proteins, it is diffusion. So and what is the requirement for diffusion? There must be a difference in concentration. There must be a high concentration on one side and a low concentration of a solute on the other side to result in diffusion. So channel proteins are passive only. They require only diffusion can take place through them. Okay. And like I said, some of these are open, but there are also gated channels, mechanically gated, voltage gated, chemically gated. So just realize these are cues that open the channel. And what you need to realize now that is gated channels are channels that are closed, and then a cue could be mechanic cue, a voltage cue, or a chemical cue that opens the channel. When the channel is closed, molecules cannot move through. But when the channels are open, now the molecules, whatever ion or so forth, can move through the channel. Okay, but we will get into these mechanically, okay, voltage and chemically gated later on. We're going to be talking about them when we talk about the nervous system, and we'll look into this more depth. But just realize right now there are channels that are constantly open. Those are the open channels, and channels that are closed unless they receive a cue, and those will be the gated channels. But again, remember, these are channels, so again, there has to be a concentration difference between the inside and the outside, because diffusion is what moves molecules through these channels. The second set of transporters are our carriers. Our carriers, we can do both passive, right? We can do diffusion through them, or we can do active transport. But the main difference is that these change conformation, and there is no direct access. It's not like there's a tunnel. It's either open to one side or open to the other side. Okay, it changes conformation. The protein changes conformation to either open to one side or to the other. Okay, and there are different types of transporters. We have uniport. A uniport, one, uni, means it only moves one molecule through. Okay? And we have co-transporters. All right? And these come of two varieties. Co-transporters move two molecules. Okay? Or it could even be three molecules, depending on, but commonly two molecules are moved through, not just one. So, and they come in two varieties. We have a symport and an antiport. Let's look at the difference between these two. Here is our symport carrier. Where do you see the movement of the molecules? Okay, they are both moving in the same direction. Where an antiport, one molecule is moving in and the other molecule is moving out. Okay, so uniport, one molecule is moving through it. Symport, two in the same direction. In antiport, two, but the molecules are going in opposite directions. All right, so now we've seen our membranes. We've gone through the two different the ion channels, the channels, and the transporters. So let's start looking at the actual movement. First we're going to take a look at facilitated diffusion. Okay, and facilitated diffusion can take place through a channel, and it can take place through a transporter. Okay? But the key is what's taking place in diffusion. This is a passive process, and diffusion means the molecule is moving from an area of high concentration to an area of low concentration. Okay, so the cell does not have to put energy in it. It is caused by the kinetic energy that is inherent in the molecules. They're going to move from an area of high concentration to an area of low concentration.
Take place both through ion channels and also through transporters or carriers. The key is diffusion. It's going to move down its concentration gradient. So there has to, for facilitated diffusion to take place, again, it's got to have a concentration gradient. That in turn means that the cell does not have to put any energy. It doesn't have to use any ATP to make that movement take place. The simplest is, looking here, through an ion channel, through a channel, where this is just open. And you can see we have larger concentration of our molecule, our triangle molecule here, and a smaller concentration here. And so facilitated diffusion will just take place through the ion channel. Okay, just go directly through. Okay, a little more complicated is the carriers, because remember carriers are going to actually have to bind the molecules. Here, with our ion transporters, the molecule just moves through that pore, moves through that little tunnel from the outside to the inside. Whereas for carriers, the molecule is actually going to have to bind. But again, this is facilitated diffusion, so for this to take place, we have to have a larger concentration on one side and lower concentration on the other. So let's take a look at this movement through the carrier. You can see the molecule comes in and the molecule is actually going to bind. And there is a conformational change in the shape of the protein, in the shape of the carrier here. Here it's open to the outside, then it's going to close up to the outside and open to the inside. Okay. And then the molecule can be released and brought inside. But again, for this to take place, for this molecule to move from here to here in facilitated diffusion, what must we have? We have to have a high concentration of this molecule out here and a low concentration out here, because again, this is facilitated diffusion. The molecule is going to move from high to low because of its kinetic energy. Okay. The difference is between the ion channels and the carriers is the molecule has to physically bind to allow opening to the inside and allow the molecule to be released and brought in. This is showing you an example of a carrier using facilitated diffusion. When you eat lots of sugar, you eat a bag of Skittles, you have lots of glucose in the blood, and then in turn there's more glucose in the interstitial fluid that surrounds the cells. So there's lots of glucose out here, high concentration, lower concentration inside the cell. These are GLUT transporters. Okay, this is facilitated diffusion though, because we're going from an area of high concentration to an area of low concentration. But what has to take place is these glucose molecules have to come in and actually physically bind. Then we get our conformational change, and now glucose can be released into the inside. Again, it is high to low. Okay, high to low. So we got high concentration to low concentration, but again, we're using a carrier or transporter, so the molecule has to physically bind. If this was a channel, but we wouldn't have a channel for glucose, but if it was an ion channel, we could just physically, right, there would be an opening here. But again, facilitated diffusion, it is going down the gradient and there is no energy being used by the cell. The energy is in the gradient itself. Alright, so that gets us through our passive movement. Our passive movement, our simple diffusion. Diffusion meaning passive, cell does not have to use energy. Now let's move on to where the cell has to use energy. And we're going to have two major forms. We're going to have primary and we're going to have secondary. So we'll look at what is required for each of these processes. But again, the cell has to use energy. And why does it need to use energy is it's going the opposite way of diffusion. Diffusion, the molecule goes from high to low concentration. Okay? A molecule does not want to go from low to high. And that is what's taking place in the active transport, is the molecules being forced from going from a low concentration to an area of high concentration. And therefore the cell has to use energy, and therefore the active portion of this. Okay, so for active transport is always going to be protein mediated. We can't have simple diffusion, molecule can't go from low to high and just directly through the membrane. Right? It is always protein mediated. It is mediated by carriers. There are no channels that allow for active transport. For active transport, it's going to be carriers or transporters that are going to be used. Okay? Again, we're going from lower to higher concentration, from low to high. So we're going against the concentration gradient. We're not going down it, we're going against it. Okay? And this actually it's going to allow the cell to create a state of disequilibrium. It's going to allow us to make concentration differences. And the cell, in some cases, is going to want a concentration difference. You can see here the little squares. There's low here, high here, and the squares are being moved in this direction. But what's required of the cell has to use some form of energy. Could come as ATP, and we're going to see with secondary, it comes from another form of energy. Okay? But again, the cell has to use energy because we're going against that concentration gradient. So let's look here at the difference between primary and secondary active transport, or you can say direct primary, indirect active transport. Okay, here is primary. Okay, the key with primary is the protein that is doing the transport, moving. You'll see X here being moved from low to high. Okay, and that allows us to create this concentration gradient. This protein is directly using ATP as its energy source. Okay, ATP is being used to push X from low to high. Okay. Secondary, on the other hand, as you see here, what's being used? All right, we're taking X We're taking X here from high to low. Okay, X is going from high to low. On the other part, we're going from low to high, but we're using X. Okay, so that is diffusion. It's going in the right direction. And there is what I haven't stated yet, but there are energy in these concentration gradients. Okay, this molecule wants to go in this direction, so that creates energy. And in this case, for secondary, the energy from X moving down its concentration gradient from high to low is going to help move that energy, is going to move S here from low to high against its gradient. Okay, so instead of coming directly from ATP, the energy being used to move the molecule against its concentration gradient, the molecule S, is using the energy from X moving from high to low. Okay, there is energy in these gradients. This movement creates energy, allowing us to move S from low to high. Okay, so it's not using ATP directly like we're using ATP directly here. Okay, in a sense, because ATP is required to make, we saw ATP was used to make this gradient. Okay, we're taking X from low to high and we're using ATP. In a sense, even though secondary doesn't directly use ATP, it is still dependent on ATP because to make this gradient for X, we have to use ATP. Okay, but the difference is this protein is using ATP directly, so this is an example of primary active transport, where this protein here is using the energy from X moving down its concentration gradient to move S against its concentration gradient. So it's not using ATP directly, so therefore it is secondary active transport. So you will see these primary active transporters. You see ATPases. Okay, those are primary active transport. They're ATPase. They're breaking down ATP to move molecules. All right, these are primary. They're using ATP to move these molecules from low to high. The one I'm going to stress this all semester, the one you guys must know is this one. We will see this one throughout the semester. This is one of the most important proteins in your body. Okay, sodium potassium ATPase, or commonly many ATPases are also known as pumps because they are pumping the ions from low to high. So this is also known as the sodium potassium pump. Okay, it is antiport. What does that mean? Antiport, remember, there are two molecules. They're co-transporters. In this case, what do you think the two molecules are? They're sodium and potassium. And they're going to move in opposite direction of each other. Okay, because remember these ATPases, what can they function? They function to actually create concentration gradients. They actually function to make an area of high concentration of one ion and an area of low concentration of an ion or a molecule even. Okay, so they are moving. In this case, sodium is getting moved out of the cell and potassium, it's moved in. Okay, but sodium potassium pump, write this one down or get this lodged in your memory. We're going to look at it in a second here, but this is one of the most important proteins in your body. About a third of your energy, the calories you eat, are used to fuel this protein, this sodium potassium pump in your body. Just showing you a visual example of, in this case it's a uniport. We got calcium, ATPase. So what do you think it's going to do? It's going to move calcium using ATP to move calcium from an area of low to an area of high. Okay, so it's going to move calcium in this way, and it is a primary transport. It's an ATPase, so therefore it is a primary transporter. Okay, it's in primary active transport. All right, so here's the coup de grace. This is the one I'm going to be harping on all semester. This is the sodium-potassium pump. Okay, it is primary active transport. It is using ATP here. Using ATP. It is found in all animal cells. So this is an important protein in biology. You find it throughout the whole animal. So basically what it is doing is maintaining concentration gradients. So remember, these pumps are used to make areas of high concentration to areas of low concentration. Okay? It is anti-port because it's going to move sodium out of the cell and potassium in. Okay? And like I said, about 30%, about a third of the ATP produced by the cells, a third of your energy used, is used to fuel this pump. Okay, so this is a very important pump, but you can see what it's doing. It's taking potassium from low to high. So it's making potassium high on the inside. And it's taking sodium out. Okay, so making sodium low on the inside, high on the outside. In turn, also, because it made potassium high, it's making potassium low on the outside. So we have two concentration gradients. We have a concentration gradient of potassium and a concentration gradient of sodium, just in opposite directions. Also, it is not an equal exchange. One pump of this molecule, we get 3 sodium molecules get moved out, where 2 potassium molecules get moved in. So it's not an equal exchange. It's anti-port, but that's not 1-to-1. It's 3 to 2. 3 out of sodium, 2 in of potassium. So I'll walk you through this, but this is kind of one example where watching videos, you guys can Google sodium potassium pump. You can actually see a video of the sodium potassium pump operating. But you can see here, what do we have? Part 1, 3 sodiums are coming in to bind. Okay, they bind, we get ATP. ATP is being used to fuel the movement of sodium from, so sodium would be what? It would be low here and high out here. So we're going to be moving the sodium against its concentration gradient. But we're using ATP, we phosphorylate the protein, and now it will open up and sodium can move out. When it's open to the outside, our friend potassium can bind. That changes, we release the phosphate, and now it opens back up to the inside. Now potassium gets moved in. Where would potassium? Potassium would be high here and low out here. So again, it is moving potassium from low to high and moving sodium from low to high, just in opposite directions. Okay. So some of the functions of the sodium potassium pump, we'll talk about this later, maintaining the resting membrane potential. But what I want you guys to really focus on right now is that the sodium potassium pump makes a sodium and a potassium gradient. We're going to use these in electrical signalings, but also you saw, what did we use for secondary active transport? We allowed a molecule went down its concentration gradient, and that energy from it going down was used to move a molecule going against its concentration gradient. So much of what this sodium-potassium pumps is, it creates a sodium concentration gradient. So now we can use that sodium can move down its concentration gradient to move other molecules. So the sodium-potassium pump fuels a lot of the secondary active transporters. So what on the secondary active transporter, what do you think the molecule that is going to be used from going from high to low is going to be? Yeah, it's going to be the sodium. The sodium has a gradient, so sodium can be used as the energy moving down its concentration gradient to move other molecules up its concentration gradient. And that's basically what we see here in this table. Okay, you can see we got most common second active transport are driven by the sodium. So in this case, sodium will go down its concentration gradient and help move these other molecules up its concentration gradient. Okay, we'll see sodium glucose, SGLT, sodium glucose. These are all secondary active transport because we're not using ATP directly, we're using the sodium gradient to move the molecule from low to high. Okay, later on in digestive, we'll see sodium amino acid cotransporters and sodium calcium transporters. Well, let's look at, so here's our secondary active transport. Here's what protein is this. If we were in lecture, I'd sit here all day and make you look at it and make you tell me exactly what this protein is, because I'm going to keep harping on it all semester, but this is what? This is your sodium potassium pump. We're using ATP, we're moving sodium out, 3 molecules of sodium out, and 2 molecules of potassium in. Again, this is active transport primary because this protein is directly using ATP. But now we have this large amount of sodium outside. And say we need to move a molecule that is inside the cell, but there's large concentration already in the cell, and there's low outside. Right? This is going against the concentration gradient. Okay? So how do we do that? Instead of using ATP in this case, we are using the concentration gradient from sodium. Sodium's going from high to low, and that energy from this gradient, from the movement of this, is going to fuel the movement of this molecule from low to high. Okay, so secondary active transport. Okay, sodium can also be used to move molecules out of the cell from low to high. But again, it is the sodium. The sodium gradient is the fuel. And how is the sodium gradient? Why do we have large amounts of sodium out here? And that goes back to the primary. The sodium potassium pump has created this large concentration gradient out here. Okay? So the sodium potassium pump fuels a lot of these transporters, among other things we will see later on in the semester. So this is a very important protein. Again, sodium potassium pump. When you see sodium moving out, potassium moving in, and you see ATP there, that's going to tell you this protein is the sodium potassium pump. So let's look at one example of secondary active transport here. Again, most of them are fueled by the sodium. Okay. Sodium, glucose, secondary active transport. Again, we're using that sodium gradient, that high concentration of sodium outside. And what we're going to be doing is moving glucose from an area of low glucose to an area of high glucose. Okay, moving sodium from low to high. And here we go. Sodium's binding, glucose binds. Okay, again, sodium, you can see sodium is high here, low here, so we're using the energy from the gradient to move glucose from low to high. Okay, binding, so we move here, glucose binds, and now we get a closing and an opening up, and now sodium and glucose can get released. So now we are bringing in glucose from low to high using the high to low gradient of using the concentration gradient of sodium. Okay, the energy is coming from this concentration gradient, therefore it is secondary active transport. So those carriers that we use for both passive and active transport will exhibit certain characteristics or properties to them. They all share. One of those is specificity, competition, and saturation. Specificity, I'm having trouble saying that word, is the ability to carry to move only one molecule or a group of different molecules. So these proteins will not just move any molecules or any ions. It will be specific for a certain ion. So therefore we have to have many different proteins for the different molecules that we have to move. Competition. Okay, members of a group of substance can compete with the other, because remember, in a transporter the molecules actually have to bind. So if there's a molecule already bound to the site on the transporter, then another molecule cannot bind there. So in the case of a glucose transport rate, we're transporting glucose. As glucose concentration goes up, you can see the rate of glucose transport goes up. This is with glucose only. Okay, again, we're looking at glucose transport. If we add galactose, another monosaccharide, glucose is a monosaccharide, galactose is a monosaccharide, so this transporter may bind to galactose. If galactose is bound where glucose is going to bind also, then the glucose can't be transported at that time. Okay, so you can see in this case the rate of glucose transport goes down because galactose is competing with glucose for transport. And then saturation, sorry this is small, but you can see as we increase the concentration of a molecule, the actual rate will go up, but at a certain point saturation occurs and the rate plateaus, because remember, the molecule has to bind. If there's only a specific amount of transporters, at a certain point, if you keep the concentration when the molecule is already bound by the transporter is already bound by a molecule, then another molecule can't bind there. Okay, so we'll slow down. It has to wait for the transporter to move the next one, and then that transporter can bind the molecule. So as we increasingly increase the concentration, it goes up, but at a certain point there's so much there that these other molecules are waiting for the transporter to release and have an open site. And so we can only increase the rate of transport so much. Okay, at a certain point it will plateau, and that's due to the saturation. And you can see it here, the rate goes up, but then the rate plateaus. Okay, all the transporters are bound And so the molecules have to wait till them to become unbound before they can be bound and transported also. So we can increase the rate up to the point of where the transporters start to get saturated. All right, we're going to finish off here. We're going to finish off moving large stuff. So remember the channels that we use for, say, facilitated diffusion? Those are just small ions. We could move somewhat larger stuff like glucose, bigger molecules with the active transport and with the carriers, and also with facilitated diffusion, but we had to use carriers. But now we want to move large items. Okay, large items in or out of the cell, and to do that we use these membrane bound vesicles to move large stuff in or out of the cell. Okay, and those are going to be endocytosis. You can think endo, in, exocytosis, we're going to exit, so move stuff out, and then a special form, phagocytosis, is going to move larger things in. Okay? So as I already said, moving stuff in, larger stuff, importing stuff in, endocytosis or phagocytosis. We'll see there is a difference between these, but again, these are both processes moving larger molecules or particles into the cell. And then exporting or leaving stuff, making stuff exit, is a process called exocytosis. But again, these are going to be using membrane-bound vesicles to move either in or out of the cell. All right, so endocytosis, we're going to move larger particles or molecules into the cell. And basically it's going to use ATP. You're going to see the cell has to move the plasma membrane. So you can see molecules moving in here. And what's going to happen is basically the plasma membrane pinches off and forms this vesicle. Basically a little plasma membrane inside the own cell itself. So these vesicles form and wrap around in basically a sphere around these molecules. Okay, so we're creating this vesicle and moving larger stuff in. Let's take a closer look at this process. Okay, many times it is receptor mediated, meaning this molecule that is going to be moved in is the receptor. Binding of it to a receptor causes this pinching of the membrane, okay, causing this pit and then the pinching off of the membrane to form the vesicle. Okay, so the molecule moves in. If it's receptor mediated, it binds the receptor. The receptor induces making the pit and then pinching off eventually of the plasma membrane to form that vesicle. Okay, bringing large stuff in. Okay, don't worry so much about the clathrin. I'm not going to go in that detail, but basically what you're doing here is moving in these larger molecules by forming this vesicle. Okay. Stuff that can be transported in, we'll see protein hormones is one way, growth factors, antibiotics, plasma membranes. These are all things that can move in, larger molecules that can be moved in by this endocytosis. But just realize endocytosis is basically we are forming this vesicle to move the larger molecule into the cell. So exocytosis is basically kind of the reverse process. We have stuff already in, okay, a vesicle, and now we can fuse the vesicle up and release whatever is there. Okay, release of your neurotransmitter is one way. The neurotransmitters will be in the vesicle. It will fuse with the membrane, and then the neurotransmitter can leave the neuron. Your cells of your endocrine system also hold, some of them hold your hormones in a vesicle, and when they need to be released, they are sent to the membrane, they dock, and then released through exocytosis. So basically the vesicle is fusing first with the membrane, then it opens up and it gets released. Okay. We can also insert proteins into the plasma membrane. Instead of this case, the proteins are just being inserted. Here, they're not actually being exocytosed, but they're being fused. Again, the vesicle is going to fuse and fuse with the membrane. There's nothing to be released in this case. Instead, there are receptors that are going to sit on the membrane. Okay, don't worry about RABs and snares. We'll get into this a little bit more when we get into the actual release of neurotransmitter or the release of hormones, and also we'll talk about the calcium in that case. But just realize for right now, exocytosis is moving out of larger items, and in this case it's the vesicle fusing with the membrane and then releasing the contents out of the cell. So since we're talking about membrane transport, kind of a good point to touch on epithelial cells. We talked about how we have to keep things homeostatic, and to do that we have to get things in and out of the body to keep the body homeostatic, can keep that internal environment homeostatic. One of those ways to do that is through transporting epithelial. Okay, transporting either stuff in or out. You can see the purple, our digestive tract. Right? What are we going to be doing in our digestive tract? We're going to be moving stuff in. We're going to be moving nutrients in to the body. So in turn, we got to do transporting stuff in and out of the cells requires membrane transport. So we'll see a lot of this transporting epithelial throughout the semesters and looking at their effects on, or the ways in which they move stuff in and out. Another one is the urinary system, right? We got to move stuff in and out, chiefly out, creating the urine. Okay, so these transporting these epithelials, the epithelials are the cells that basically are our interface with the external environment. So they are going to many times have to do membrane transport because they have to move stuff in or out of the body. Looking closer at these cells, many of the transport epithelial, they have these tight junctions. Okay, if you remember, there's not much extracellular fluid between the cells. They are basically, epithelials are cells attached to cells attached to cells. Okay? And many times they have these tight junctions. Now materials can't move between the cells when these tight junctions are here. So that forces, if anything needs to come through the body, it has to move either in to the epithelial or out to the transporting epithelial. Okay, so therefore we must have membrane transport taking place. Okay, also to note on these epithelials is they'll have two sides. Okay, the apical side, that faces, in this case, the external environment, and then the basolateral side faces the extracellular fluid. So if we move something from here into here, we're essentially moving it from the external environment into the body, or vice versa. We can move from the body to the external environment. Continue looking. And many times these transporting epithelials are what we call polarized. Okay, they'll have different transporters on different sides. Okay, the apical side or the basolateral side. And that allows for either absorption, moving from the outside into the body, or secretion, going from the inside, from the extracellular fluid, to the outside, to the lumen. And there are some epithelial cells in which there aren't tight junctions, and so there are ways in which molecules can move between the cells, and that would be what we call paracellular transport or path. And then those that are forced to move in or out through the cells, that is the transcellular pathway. Okay, and many times this pathway, the transcellular pathway, requires different transporters or channels for movement of the molecules in or out of the cells. So this is by far probably one of my favorite slides of the lecture, because this basically puts it all together. We're looking at ability of transport of glucose. And if we were in class, I'd have you talking to your neighbor, looking at which is facilitated diffusion, secondary active transport. But I'll just walk you through this, and this shows you kind of the polarizing of the sides. So we look at here, we start here, we're looking at lumen, we're bringing, what ultimately we're trying to do is bring glucose into the body. So how are we doing this? Glucose is high in the cell, but is low out here in the lumen. Okay, this we're looking at the kidneys. We're basically, if we filter out glucose, we bring the kidneys bring the glucose back into the body so we don't lose it. But here, glucose is moving from low to high. All right, so glucose is not moving by diffusion, right? It's going against its concentration gradient. But what is sodium doing? Sodium is going from high to low. Right? Remember, many of our secondary transporters utilize the energy in the sodium concentration gradient. And so what does this, number one, look like? What kind of transport would you be talking about? We're using the sodium gradient, the energy there, to move this glucose from against its concentration gradient from low to high. And therefore, you would find, right, secondary active transport. Secondary. Now let's follow glucose this direction through protein number 2. Glucose is now high and glucose is low in the extracellular fluid. So we're moving from an area of high to low. What's that tell you? Right? It's diffusion, right? Can glucose move directly through the membrane? No, because it is a larger polar molecule. So glucose is moving from high to low. It's doing diffusion through a protein. So now we have facilitated diffusion taking place. Now let's look at our friend sodium. Sodium is we need to keep this sodium low because we need to go from high to low. If sodium keeps coming in and this builds up to high, is there any more concentration gradient? No, we would lose the gradient here. Okay, so to keep sodium high and keep this low, we have to move the sodium out. And so how are we doing that? Sodium is getting pushed out here. It's going from low to high, so it's going against its concentration gradient. But unlike how we used here, we used a sodium gradient to move glucose. Instead, we're moving sodium out by using ATP. Again, we're going from low to high, so this can't be diffusion. This has to be active transport, and we're using ATP. So this is primary active transport. Who else do you see involved in here? We're bringing sodium out and potassium in. So I know I've harped on this throughout the lecture, but what protein is this then? Can you name me this protein? Yeah, it is the sodium potassium pump. Okay, you will be seeing this protein throughout the semester, the sodium potassium pump. So make sure you get knowledgeable on it. Okay, but sodium potassium pump, we use ATP to pump this out, and that allows us to keep sodium low inside the cell and allows us to continually use this secondary active transport. Okay, because if there's no concentration gradient, there's no energy to move glucose in, and so glucose will not be brought in, and you'll lose it. Basically, in this case, you would lose it in your urine, which should not be happening. Okay. But I do like the slide. Definitely look over the slide, because this is one of those important. It basically puts all that we talked about in this lecture. It puts it all together. Okay.